Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. John Mann will present Brain Plasticity, the Effects of Antidepressants on Major Depression. The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, and schizophrenia. Since 1987, the Foundation has awarded more than $394 million to fund more than 5,700 grants to more than 4,700 scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists. I'm delighted to introduce Dr. John Mann. Dr. Mann is the Paul Janssen Professor of Translational Neuroscience and Psychiatry and Radiology at Columbia University Medical Center and Director of Research and Molecular Imaging at the New York State Psychiatric Institute. He is a member of our scientific council and was a 2008 Foundation Distinguished Investigator grantee. Today's webinar will begin with Dr. Mann's presentation. This will be followed by a question and answer period. To submit your questions, please use the questions tab on the control panel on your screen. Feel free to submit your questions throughout the presentation. I'll present as many of those questions to Dr. Mann after his formal presentation. So now I'm pleased to introduce Dr. Mann. John, the floor is yours.
Thank you. Good afternoon. I'm just getting these slides organized so that you can all see them.
Excellent.
Okay. So today we're going to talk about neuroplasticity, or the plasticity of the brain. And we're going to try and understand, first of all, how some of the original antidepressants, which are still the frontline antidepressants like SSRIs, work and fit in with what we now know about brain plasticity and depression. And then we're going to switch from those medications to the new frontier of antidepressant treatment as exemplified by ketamine, and see how that fits in with brain plasticity in terms of its mechanism of action. Now, the goal of doing this is to sort of give you a better sense of how these medications work and methods that we're sort of using to try and identify newer, better, faster acting antidepressants for the future. Everything I'm going to tell you today has been funded by either the National Institute of Mental Health, NIMH, or by BBRF. As a matter of fact, the award that I got a few years ago is actually going to be featured in part of this talk. I do receive royalties from the New York State Research Foundation of Mental Hygiene for commercial use of the Columbia Suicide Severity Rating Scale. But I want you to know that any doctor, academic institution, clinic, etc., can use this scale for free in their work. In the scope of this lecture, we're going to address whether we can learn something about how or why certain medications or antidepressants from our knowledge of the neurobiology of major depression. This is how medicine functions in general. you find out what's biologically wrong, that's the basis of the illness, and then you design treatments to fix that problem. But in psychiatry, we've discovered the medications mostly by accident, and then figured out their properties, and then try to work backwards from their properties to what's wrong with the patient and what's the biology and the pathology of the depression. But in fact, and that's been the case now for 30 or 40 years. But in recent times, we've actually learned more about the actual pathogenesis of depression and what's wrong biologically that causes these kinds of illnesses, and then gone back and taken a look at how these antidepressants work to see how the two fit together. And this lecture is going to sort of walk you through that story. Now, most of the pharmacotherapy for major depressive disorders has been classified according to its initial property and how it's worked, in terms of what we've known about how it's worked. So, for example, if you take MDD, Major Depressive Disorder, there's a list of medications there, and they were kind of discovered as effective for that condition, and that's why they've been called antidepressants. But other medications that can help depression were discovered for other indications. For example, new generation antipsychotics that you see in the middle of this list, NGAs. New generation antipsychotics are antipsychotics. And as a side observation, after these medications were developed and tested for psychosis, and then as anti-manic drugs, it was discovered that some of them had some antidepressant properties. So we've added them to this list. But our system for naming these medications really comes from the first thing they were found to be useful for. So that's why NGAs are called new generation antipsychotics, and so on. Okay, but the real way to go about understanding medications and how they work is to think about how their pharmacological targets of action fits with the known neurobiology of the illness that they're used for, in this case, major depressive episodes. There are many ideas about what causes major depression. I've listed here 6 pathways, and this is an incomplete list, but it does capture the main currently in vogue models and ideas about the causes of depression. I'm going to focus on the 3 pathways that are here in red. One relates to serotonin, one relates to the neurotransmitter GABA, gamma-aminobutyric acid, and the third one relates to glutamate, another major neurotransmitter. But there are other theories. One relates to the noradrenergic system, one relates to the HPA axis and cortisol overactivity, and another one relates to some kind of interactive combination of polyunsaturated fatty acids, which are known as PUFAs, and their interrelationship with neuroinflammation. But those are topics for another day. Now let's start with serotonin. This is where some of the earliest work in depression began, and people observed that when they looked at the metabolite of serotonin, 5-hydroxyindolacetic acid, also known as 5-HIAA, that in cerebrospinal fluid, which is a way of kind of getting a biological measure of things going on in the brain, there seemed to be low levels in people who were depressed. One of the things that would cause low levels of CSF 5-HIAA would be that there's less serotonin being released in the brain. Every time you release serotonin in the brain, a little bit is broken down into 5-HIAA. So the more serotonin is being released, the higher the 5-HIAA, and the less serotonin that's being released, the lower the 5-HIAA. So this led to the notion that there's less serotonin being released in the brain, and it produced this hypothesis of a serotonin deficiency. And for many years, right up to the present day, doctors have told their patients, and the literature has told patients and their families, that a serotonin deficiency is one of the characteristics of major depression. As it turned out, this was only half correct. There is a deficiency in serotonin release in the brain of people who are depressed, but it is completely due to a completely different cause to the one that most people thought. What this slide shows you is a study that we've done where we looked in the brain of people who were depressed. And that's shown by a typical case from a person who died by suicide when they were depressed. So we got some brain tissue from them, just like people get organ donations from deceased loved ones through the permission of the family. And we compared that brain, or actually compared a whole group of brains, but I've chosen typical examples, to a control. The control was somebody who had no psychiatric illness and died suddenly of something like a heart attack or in a motor vehicle accident. And we stained the brain for serotonin neurons, which are these black blobs that you can see here in the picture. So this black blob here, and this one, these are all serotonin neurons. And then there are processes that come off the neurons, which are these lines that you see in between. All of them are stained for a serotonin marker. And what you can see immediately that's different between the two groups is that the suicide depressed person has more serotonin neurons than the control, and that even the processes are darker than the control, as well as the cell bodies. So these cell bodies look darker than these ones, and the processes show up more boldly. This means we have stained this tissue for the main enzyme that makes serotonin. And then we now know by quantifying these kinds of slides that there is more capacity to make serotonin and per neuron and more neurons in the brain of people who've died with depression than in people who do not have a psychiatric disorder. So just looking at the brain at the level of the neurons, there's plenty of serotonin. So what is going wrong? In this picture, I have tried to illustrate the anatomy of the serotonin system in a very simple way. I have shown here the serotonin neuron in yellow. And at the left side of the screen, over here, is the cell body. And this is one of those processes or lines that you see. And this is a serotonin nerve terminal, and it's ending up here on some target neuron elsewhere in the brain, let's say in the cortex somewhere. Now, when the neuron fires, which it does at the cell body level, the action potential or electronic signal travels all the way down the neuron to the very end, the terminal, and causes the release of serotonin, and serotonin acts on these things here, which are called receptors. And that's the serotonin signal. So now if we look over here, we know there's plenty of serotonin. But if we look over here, there's a deficiency of serotonin. So somehow the serotonin is not being released. Now, at this end of the neuron, there is a feedback system to shut off serotonin firing, because the brain over here doesn't like there to be too much serotonin release, and it also doesn't like it when there's a deficit in serotonin release. So it does its best to fix this and keep the signaling in the right zone. Too much serotonin can actually make people psychotic. Not enough serotonin, and they get depressed. You want it somewhere in the middle. It's like Goldilocks. Not too hot, not too cold, somewhere in the middle. One way of keeping things in the middle is to control the rate of serotonin firing. Every time the brain fires, the neuron fires, a bit of serotonin is released backwards over here on what's called an autoreceptor, and that shuts off the serotonin firing. So we don't want too much release, so we shut it off if there's too much being released more quickly. So this is a very finely tuned system, and there are other kind of feedback systems built in here that I haven't shown you for simplicity's sake, but this is a crucial one. Now, here's a very interesting, I'm going to now tell you the story out of sequence. Because of some observations we made about the serotonin 5-HT1A autoreceptor that shuts off serotonin firing when serotonin is released, so that you don't have too much firing and too much serotonin being released, colleagues of ours at Columbia made a mouse in two flavors. One flavor where it has the right amount of autoreceptors, and a second flavor where it has a high number of autoreceptors. So you have a low autoreceptor mouse and a high autoreceptor mouse. And the high autoreceptor mouse was found to result in, it's shown here in the gray, less firing. This thing is the firing rate along here from 0 to 14. And you can see most of the firing is 4 and under. This is the low autoreceptor mouse with a not as good feedback system. And you can see there's plenty of firing right up to 12, which you don't see anything like that over here. And then if you measure, as they did, the amount of serotonin being released, not surprisingly, the high autoreceptor mouse with its super efficient feedback autoreceptor system has less serotonin being released. So now we can see that this may be potentially relevant to our situation. Now, when they examined these two flavors of mouse in two different tests of depression, one where the mouse swims, is put in a beaker and swims like mad, and you measure how long it takes for the mouse to give up and stop swimming. And the second one where you hold the mouse up by its tail, so it's suspended by its tail, and that's called the tail suspension test. And the mouse will struggle, and then after a while will give up. The giving up is a sign of depression. And the mice that are prone to depression give up more quickly. So the period of active swimming or the period of active struggling in the tail suspension test is shorter in those mice. Well, very interestingly, the mouse with the super efficient feedback system, because they had more autoreceptors, the low serotonin firing, low serotonin release, high autoreceptor mouse, that mouse on these two tests comes out as depressed compared to the other mouse. Okay, keep that in mind. And now these findings, which we made before the mouse was invented, will suddenly become very instructive. we looked in the brain of our depressed suicides. And sure enough, we looked at these autoreceptors in the brain of these suicides, and the number of these autoreceptors in the depressed suicides compared to the controls was much higher, which would mean that the depressed suicides biologically have a super functioning high autoreceptor feedback system. And from the mice, we understand that would mean less serotonin neuron firing and less serotonin release. So that effectively negates the fact that they've got plenty of serotonin production capacity and plenty of serotonin stored up, because they're just not releasing it. It's like having a water supply, but the pipe that's supposed to deliver the water is very narrow and very little water is getting through. So you effectively have a water shortage despite plenty of water in the storage system. Now, using an imaging approach called positron emission tomography, or PET scanning, PET, PET scanning, we can actually quantify these autoreceptors in the brain of patients. And that's what this shows you. These are pictures of the brain, top down, front to back, and side on, showing these 5-HT1A receptors. The ones that are located here, where this arrow is pointing, are in the position of the upper brainstem, the midbrain, dorsal part of the midbrain, which is where the serotonin neuron cell bodies are located in the brain. They're not all over the brain. They're only clustered in that part of the brain stem. And then they project up to the rest of the brain. So these other receptors, they're the receptors at the nerve terminals, but they're not the autoreceptors. The autoreceptors are just here on top of this green arrow. So we can quantify that, and we've done that in depressed patients. And when you do that, you'll see here, which is the results of group averages for the PET scans in different groups. The more red the color, the more receptors there are. I want you to focus on the autoreceptors, which are located here in this picture. These are the controls. These are the, NRM means not recently medicated, depressed patients with his diagnosis of MDD. And then down the bottom is what happens when you mathematically subtract the binding of the controls from not recently medicated, and you see a positive result. In other words, there's a deficiency, there's an excess of autoreceptors in live, unmedicated, untreated, depressed patients. So this is the same as what we saw in the postmortem brains, and it shows you that this abnormality is also present in depressed patients. Now, we also looked in depressed, unmedicated patients who had bipolar disorder, and they show the same excess of 5-HT1 autoreceptor binding. So whether the major depressive episodes are part of MDD or whether they're part of bipolar disorder, they share this characteristic of more autoreceptors. So that means that this mouse model, biologically and behaviorally, has some significant resemblance to the patients. Now, in order to determine whether this was only present when people were depressed or also present between episodes of depression, because depression is an unusual illness in that it is characterized by being episodic for most patients. Not all patients, but most patients. Most patients have an episode of depression, and in between they feel well. Some people have episodes of depression. In between they feel less depressed, but they're never really free from the depression. But for a lot of patients, it's episodic. But when they're well, they think, they're completely cured. That's how it feels anyway. And the doctor has to give the patient the bad news that, if you've had a couple of these episodes in the past, the odds are that you're going to have another episode in the future unless we do something to prevent it. So this study shows you that, in fact, when the patients were remitted, which is shown in the blue, compared to currently depressed, which is shown in the orange, in many brain regions, but particularly I want you to focus on this section that says RN, which is the part of the brain where those autoreceptors are located. Even when the patient is better, they're still showing elevated binding compared to controls or healthy volunteers whose binding is shown in the gray. So this elevated autoreceptor binding is a biological trait that's present during and between episodes and possibly explains why people have recurrent episodes of depression, because biologically this part of their brain actually doesn't recover and normalize even though they feel better. Now, about 30 years ago, 20 to 30 years ago, a long time before we found all of these things, which we've only found recently, in the last decade or so, studies in rats and mice by two Canadian investigators called Claude DiMontigny and Pierre Blier were looking at SSRIs and how they worked. And they were trying to solve the problem, why is it that SSRIs take weeks to work? Why don't they work right away? After all, they worked by blocking the serotonin transporter, which increases the amount of serotonin in the synapse. So signaling should be better. And they do that right away. So why don't people feel a lot better after a couple of doses? So when they studied the action of SSRIs, they discovered that the SSRIs produce an increase in 5-HT serotonin initially, but almost immediately that firing rate is shut down and the release rate of serotonin drops right off. And it takes weeks for this to gradually reverse and the firing rate to come back to normal, and then the amplification of the serotonin release in terms of signaling by blocking its reuptake into the serotonin neuron begins to become effective in terms of amplifying the signal. In other words, SSRIs depend on the serotonin being released, because all they do is, after the serotonin is released, they slow down its removal from the synapse by the transporter, amplifying the serotonin signal. But if the serotonin firing and release drops off and takes weeks to return to normal, the benefit of the SSRI takes weeks to appear. Now, they eventually figured out that this was due to the fact that the SSRI, the serotonin, immediately shut down the firing of serotonin neurons through these 5-HT1A autoreceptors. And one of the interesting effects of SSRIs over many weeks in these laboratory experiments in mice and rats was that it reduced the function and number of these autoreceptors slowly over weeks. And as it did that, the firing picked up and the amplification of the serotonin signal got better and better. So they figured this out a long time ago, but at that point, nobody had any idea really what was going on with the 5-HT1A receptor in the brain of depressed patients until we did these experiments. Now, 5-HT1A autoreceptor binding and treatment outcomes are really a very interesting question in terms of patients. And no one had really managed to study this until PET scanning became available, and one could do such a study in patients to see if what happened in the animal experiments of DiMontigny and Blier also happens in our patients. So we did a study in 24 unmedicated patients with MDD following 8 weeks, average of 8 weeks of standardized pharmacotherapy with escitalopram and SSRI. And here's what the patients looked like. We divided them into 2 groups, remitters and non-remitters. And you can see that at baseline, the 2 groups were very similarly depressed in terms of severity, no real difference. The suicide attempt rate was fairly similar in both groups. Well, there's a few more attempters in the non-remitters, so that group didn't do as well. But the groups at baseline pretty much had the same kind of severity of illness. The remission rate was pretty good in our study. 46% of the patients remitted. And here's what happened with the PET scan. This shows you the same thing that you're now used to. Here are the patients before treatment, and here are the same patients after treatment. And you can see that if you focus on the degree of binding here in the autoreceptor, that this gets smaller when you go over here. And in fact, there was an 18% decrease in the autoreceptors after the SSRI treatment. And we did the scans on an average of 7 weeks after they were treated. So they had a robust response, and they had a distinct reduction in the number of 5-HT1A autoreceptors, which was very exciting because this suggested that this autoreceptor reduction, which was seen in the animal experiments, is also part of what happens in patients, and it could be a way of tracking if the antidepressant is really doing its job biologically and not just clinically. Now, I promised to link this all to neuroplasticity. So now let's look at neuroplasticity briefly, again in mice. These are my colleagues at Columbia who work with the mice, Dronovsky and Renee Henn. And this is the part of the brain called the dentate gyrus. It's shown here in a stylistic diagram. Dentate, this is the brain, and here is the dentate gyrus shown here. And this is a part of the brain in the hippocampus, which in people, human beings, and in mice, there is ongoing neural plasticity with processes getting bigger and with neurons multiplying and not multiplying. So in the brain, if you subject mice to chronic stress of different types, you actually see an effect in terms of a loss of the neurons in the dentate gyrus, which is shown here by the fewer red dots. You also see that the processes, which are here, this is the control mouse, get smaller. So there's a loss of synapses and contacts, and this is a general loss of functionality in the brain, which you can be seen very clearly here in the dentate gyrus. However, if you stress the mouse, which makes it depressed, and produces these biological changes. And at the same time you give the mouse antidepressants, you can actually prevent this happening. That's what these other pictures show, that you can actually prevent it happening. Stress plus the antidepressant, and the mouse looks pretty much normal. And in fact, you can actually produce, with the antidepressant alone, an overshoot, more neurons than they actually would have otherwise, a kind of compensatory overshoot. But basically, you reverse the depressogenic biological effect of the stress with antidepressants. Now, there's some clue that this may be happening in people. This was a study that was more than 20 years ago, looking at the hippocampus in depressed patients by Yvette and colleagues, and they showed that the more time the patient spent in a depression, the smaller the hippocampus. Now, this doesn't prove cause and effect, but it suggests that being depressed for a longer period of time means that you may end up with your hippocampus smaller, which, according to the mouse data, would be consistent with the idea that the processes and the synapses are getting fewer, and that the neurons are being lost over time when the person spends time in a depression. So why would the brain be smaller in major depression? Loss of neurons, fewer synapses, and maybe other elements of brain tissue, such as glia, which are non-neuronal structural tissue cells, and vascular tissue, which the brain needs in order to provide oxygen and remove waste products and stuff like that. Now, we have looked this is work mostly by my colleague in our division, Mara Baldrini. We looked at our postmortem brain collection, and we looked at the volume of the hippocampus, and that's what this picture shows you. Here's the dentate gyrus that we focused on, because this is the most brain plastic area in the hippocampus, and we outlined the dentate gyrus and measured its volume. And when you do that, here are controls. This is the volume of the dentate gyrus in control. Here are untreated depressed people. They were depressed at the time of that they died, but they were not being treated with antidepressants. And these are depressed people who at the time of death were on SSRIs. And lo and behold, they look just like the controls. And these people have about half the volume of the dentate gyrus. So if you believe the mouse data, then you would say that this may represent the loss of hippocampal or dentate gyrus volume due to the depression, and this may represent the correction of that abnormality with SSRI treatment. Now remember, I suggested that one of the causes of this loss of volume in the dentate gyrus might be fewer neurons. So we looked at the number of mature neurons, they're called granule cells, in the dentate gyrus in untreated depressed people. And we found that these are the controls. Here's the number of neurons. Here are the number of neurons in the untreated depressed. And here are the number of neurons in the SSRI treatment group. So you see exactly the same pattern, a significant deficit of neurons, mature neurons, in the untreated depressed, and the treated depressed on SSRIs look the same as the controls. It seems like good news for SSRIs. How do you get to that position? Well, we also looked at the progenitor cells. In this part of the brain, as I mentioned earlier, the dentate gyrus, this is one of the very few places in the brain that in adult human beings, new neurons are being produced at a steady rate. But when you give SSRIs, you produce a dramatic increase in the number of these progenitor cells multiplying and maturing to neurons in the mouse. And when you count these progenitor cells that are multiplying and presumably on their path to become new neurons, here are the controls, here are the depressed, not very much. There's some activity, but it's not very robust. But there is some activity in both groups. Looks a little less in the depressed compared to the controls. But look at this whoppingly great number in the depressed on SSRIs. So one way of thinking about this is a kind of catch-up. This is a catch-up process whereby SSRIs in adult humans with depression produce a big increase in activity in these progenitor cells in order to produce more neurons, in order to correct the deficit in neurons that you might be seeing in untreated depression. Now, what's fascinating is to connect all of that information with the 5-HT1A receptors that we were talking about before. Not the same ones in terms of being autoreceptors, but remember that you saw there were 5-HT1A receptors, if you think back to those pictures, in other parts of the brain. Those are on other neurons, the targets of serotonin. They have a different function and a different role. One role that they have is revealed by this mouse study that you see here, again from my colleagues here at Columbia. If you take an SSRI and give it to a mouse and want to produce an antidepressant effect, you need to have these 1A receptors on these neurons in the dentate gyrus. Because if you do not have those receptors on those neurons in the dentate gyrus, you do not get an antidepressant effect. And here's the evidence. This is called vehicle, which means giving something other than fluoxetine or Prozac. And this is giving fluoxetine F or Prozac. So when you give that to mice that are swimming, you see that the amount of time spent not swimming goes down dramatically. In other words, this is a big antidepressant effect in those mice. But if you knock out this 1A receptor on those dentate gyrus neurons, which incidentally also blocks the neurogenesis effect, you will also knock out the antidepressant effect. So if you really wondered whether the antidepressant effect depends on this neurogenesis effect via these 5-HT1A receptors on those very neurons that I showed you were deficient in the human depression, this experiment shows that those receptors are absolutely crucial. And those receptors are needed for both the antidepressant and the neurogenesis effect. Okay. Now, in human beings, we have, and others have shown that the amount of gray matter throughout the brain in healthy people is proportional to the amount of these 5-HT1A receptors on in those brain regions. It's not just dentate gyrus, but other brain regions where it doesn't play a role in neurogenesis, but it does play a role in those other brain regions on synaptogenesis and processes. So there is a lot of human data that supports this relationship. For the last part of our time together, I'm going to talk a bit about GABA and glutamate. GABA. There is a lot of evidence from different types of studies that a GABA deficiency is present in major depression. There are fewer GABA neurons post-mortem in both bipolar disorder and probably MDD. And when you measure GABA with spectroscopy, in the occipital cortex in depressed patients, there is a deficiency. And we've shown a relationship between CSF GABA and the severity of anxiety and depression, because GABA plays a role in anxiety as well as depression. Now, what can cause a fewer GABAergic neurons? No one is absolutely sure, but some people have speculated it's due to a lack of trophic effects via 5-HT1A, serotonin, and BDNF, brain-derived neurotrophic factor, or toxic effects due to things like too much glutamate, which I'm going to discuss in a moment, or an excessive amount of glucocorticoids, like cortisol due to HPA axis overactivity. So, but what's interesting is that many of people don't realize that GABA has been a therapeutic target for antidepressants in mood disorders for a long time. For example, anti-epileptic drugs, which are used as mood stabilizers, and some may be antidepressants, work by increasing GABAergic transmission. That's why they were developed as anti-epileptic drugs, because GABA is an inhibitory neurotransmitter, mostly in the cortex, and it inhibits the hyperexcitability of the brain and epileptic foci to prevent epileptic seizures. But in terms of depression, which is potentially a GABAergic deficit disorder, it may also work to stabilize mood and have some antidepressant properties. One of the most potent known antidepressants is ECT, electroconvulsive therapy, and that one of its most distinctive effects is to raise seizure thresholds, and it does that through the GABAergic system, at least in part. And finally, ketamine, which I'm going to tell you about next, raises GABA levels in the anterior cingulate cortex. The two commonest neurotransmitters in the cortex, in the brain, are glutamate and GABA. GABA tends to be an inhibitory neurotransmitter. Glutamate tends to be an excitatory neurotransmitter. There's some evidence that there's a glutamatergic excess in major depression. Now, we do know that some glutamate is good for you, and it causes long-term potentiation, which is fundamental to memory formation. In other words, it causes new synapses to form, and that's how we form, we sort of encode memory long-term. But too much glutamate is also thought to be potentially toxic. Now, ketamine is a fast-acting antidepressant and enhances glutamate levels, and that raises questions about how does it work. If too much glutamate is already present in depression and ketamine raises glutamate, how can it possibly be a useful therapeutic? Well, glial cells, the non-neuronal cells in the brain, which act as a kind of brain structural cell, have one of their actions is to remove glutamate from synapses. So they kind of clean up and suck up the glutamate. And if you have a deficiency in glutamate removal, you will have an excess of glutamate, which is bad for you. If you have a loss of glial cells, which has been reported in the cortex in studies of depression post-mortem, you may be vulnerable to too much glutamate happening. So impaired uptake of glutamate by glial could lead to toxicity and neuronal loss, and this excitatory toxic effect may be mediated via the so-called NMDA receptor. So how can glutamate NMDA receptor signaling, therefore, be the target of action of drugs like ketamine, which is well known to be an NMDA and a receptor binding drug? And the answer is ketamine does bind to the glutamate and NMDA receptor, but as an antagonist. It blocks it. Now, we have ways using PET scanning of imaging the amount of glutamate in the brain. here's a study which shows you what happens when you enhance glutamate transport, which means glutamate removal in the brain. And this was done with a drug called ceftriaxone. And we used a PET tracer, or somebody used a PET tracer called ABP688. And ABP688 binds to a receptor that glutamate binds to. When you have more glutamate, you'll have less binding of this PET tracer because the glutamate prevents it binding. So it's a kind of mirror image. More binding of the PET tracer means less glutamate is around. And when there's more glutamate, it prevents the PET tracer binding, so there's less PET tracer signal. So these are mice before, or rats rather, before anything's done, and there's some binding of this PET tracer in different parts of the brain in the mouse. And this is the binding after giving this drug, ceftriaxone, and increasing the amount of uptake of glutamate, lowering its levels. Lower the glutamate, increase the amount of binding. That's exactly what you see. Now, here's a study that was conducted looking at the amount of this binding in depressed, untreated patients, showing that the depressed patients have less binding than the healthy people, which means if they have less binding on the PET scanner, there's more glutamate in their brain because that's preventing the binding. So this means that there's an excess of glutamate in depressed patients. We did a study using a different approach. We used, and this was the study that was funded by NARSAD initially, in the pilot study. That's how we proved that this really works. We gave depressed patients an infusion of ketamine as a treatment. We put them in the scanner. They went in the scanner depressed. While they were in the scanner, they got ketamine. And they came out of the scanner feeling cheerful. And we scanned them all through that time to see what happens to GABA and glutamate during the ketamine infusion in depressed patients during treatment. And here's what we found. Well, this is how we did it. We looked at this bit of the brain here, which is over here, which is a key part of the brain that's involved in depression, using magnetic resonance spectroscopy. And here's what the results show. This is the ketamine infusion. This was funded by the foundation. Here is GABA, and there's a rise in the GABA levels, which has gone back to baseline by the end of the infusion. And there's a rise in the amount of glutamate, which we measured in this peak called GLX. So both of these neurotransmitters get a big bump when people get ketamine. And what does that mean? Well, we also looked at another antidepressant, which also works by blocking the NMDA receptor, and we found also an increase in GABA and glutamate. Green is GABA, orange is glutamate, GLX, and both show a clear rise as a result of treatment with this drug. And both drugs, ketamine and this GLX, have antidepressant effects. So this is not an effect related to ketamine, but other drugs that block the NMDA receptor may be antidepressant. So we then did a study looking at different doses of ketamine, including no placebo, and we found that just like, I've shown you this already, we found that ketamine produces a dose-dependent reduction in depression severity, and this effect is mediated by this increase in glutamate, which is very interesting. Now, using PET scanning and the other method for measuring the effect of ketamine on glutamate binding, other people have found that ketamine produces a reduction of this ABP PET tracer binding, which means if the binding goes down with the treatment of ketamine in this PET study, which is completely different to our MRS study, they also found a glutamate surge. So now we have two different studies that replicate our findings. How does this work? So we know that ketamine produces a rapid and robust response in depression and even in suicidal ideation. This has been shown in several clinical controlled trials, and the benefit persists for a while. And what we think happens is this: you get a surge of ketamine in people of glutamate in people who already have plenty of glutamate, but you have blocked the NMDA receptor. So this extra ketamine effect and the glutamate effect on glutamate is now channeled through other receptors. Another family of receptors are called AMPA receptors. These AMPA receptors don't have a toxic effect. They make more synapses. They do that rapidly in hours. And that fits much better with the time frame of people feeling much better in 24 hours, which is the time that we were measuring the antidepressant effect as an outcome in our ketamine studies. At 24 hours, people are substantially better. At 24 hours, the downstream effect of this glutamate surge produced by ketamine is the production of a lot more synapses. That synapse formation, we believe, is fundamental to the antidepressant effect of ketamine and other drugs like it. What's fascinating is that if you go back and look in mice, you not only produce an antidepressant effect in mice, but you can even make the mouse 2 months later resilient to the same stressor effects that produced from the chronic unpredictable stress that produced depression in the mouse in the forced swim test. 2 months later, after the ketamine, they still remain resilient. You can make people more resilient, potentially, this hasn't been tested in people, by giving them ketamine. It not only might have an acute antidepressant effect, it may have a longer-term resilience effect. So just to summarize what we have discussed today, you've seen that depression pathogenesis involves impaired serotonin release due to autoreceptor overexpression. This chronic lack of serotonin release can lead to brain atrophy, particularly in critical areas like the dentate gyrus and the hippocampus, but also in other brain areas. Antidepressants can correct this autoreceptor overexpression and produce an increase in serotonin firing and presumably a serotonin-mediated trophic effect on the brain. Now, ketamine also produces a trophic effect on the brain through a completely different mechanism. It switches glutamate transmission from the NMDA receptor, where it may be potentially toxic since they already have an excess of glutamate when they're depressed, to the AMPA receptor, which is the trophic receptor, producing more synapses. And this trophic effect is what we believe is antidepressant. And because ketamine works through a completely different mechanism to SSRIs, it may explain why ketamine works as an effective antidepressant in many patients whose depression has not responded to SSRIs. And this is the group of individuals whose work I've just had an opportunity to tell you about. Thank you.
John, thank you for really an extraordinary presentation. And I think it shows how important it is to understand the basic mechanisms and how that ultimately can lead to a better understanding of the brain, a better understanding of the illnesses, and then new treatments. We only have time for one question, and I want to ask you about the issue of ketamine and helping people who have refractory depression get better, but also the issue of the how long the ketamine works, and the fact that people respond right away, but that response may wear off. And I'm curious how everything that you just explained today, how that might fit into the use of ketamine or a similar type of medication for ongoing treatment for somebody who has refractory depression.
That's a terrific question. So in our studies, we have only used 1 or 2 doses of ketamine. We published a study. Mike Grunbaum was the first author, was in the American Journal of Psychiatry last year. And in that study, we took people who were already on antidepressants, and we just gave them the ketamine. Their depression wasn't getting better. And they had a meaningful antidepressant response, and they were selected because they were very suicidal, and they had a terrific improvement in their suicidal ideation. But the important thing about that study is that we followed them. We gave them optimized psychopharmacology with antidepressant medications, oral medications after the ketamine. And whether it was the group that got ketamine immediately or the group that first got a placebo and then got ketamine after that, both groups were doing just about as well at the end of 6 weeks as they did after the ketamine. In other words, with optimal antidepressant treatment being administered at the same time or immediately as soon as the ketamine is given, patients do really, really very well. This idea that they're just going to slide back into a depression doesn't appear to be true based on our findings. Now, other people have tried different approaches, for example, giving a couple of doses of ketamine a week for several weeks and then reducing it to once a week and then once every 2 weeks and then once a month. And that seems to work pretty well. If the patient doesn't get it better, by the way, after 2 doses of ketamine, there's no point in going on trying because they're not going to respond. They're just not a responder. But if they do get a very good response from the first or second dose, then you've got a choice. You can either just continue or optimize the antidepressant medications, or you can give them more doses of ketamine. We know a lot less about the best way to dose the ketamine, if it's multiple doses, and the safety and longer-term efficacy of that approach than we do about the single dose.
Well, thank you. That's very important information, and I have a feeling that in the not-too-distant future, you and others will be able to present more about that and really have an impact on many people's lives. John, I want to thank you for this presentation, for the extraordinary work that you've done over the years and continue to do, for being a member of our scientific council. So thank you very, very much. I also want to thank everybody who joined us today. All of the research we fund is made possible through private donations, so please consider making a contribution by visiting our website, bbrfoundation.org, or calling us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion or would like to share it with a family member or a friend, please visit the events and webinar page on our website. I hope you'll join us again next month when Dr. Jeff Lieberman, Chairman of the Department of Psychiatry at Columbia University College of Physicians and Surgeons, Director of the New York State Psychiatric Institute, and Psychiatrist-in-Chief at New York Presbyterian Hospital, will present Early Detection and Prevention of Psychotic Disorders, Ready for Prime Time, with a question mark. The webinar will take place on Tuesday, February 12th at 2 p.m. Eastern Time. Once again, thank you for joining us and enjoy the rest of your day. Take care.